Welcome to my channel. This is Moody. So today's lesson I'll be teaching you how a cell does what? Transport things across the cell membrane, both in prokaryotes as well in what? Eukaryotes. So you need to understand that it is very important to understand this because it will give you reasons why certain things are not allowed to pass through the cell membrane, or why certain things have to pass through the cell membrane using those means which they use. So for you to understand this, take this as your cell. Then if you are to cut it at that point, also that point, then it will look like this, and those are the things which are what? Associated with what? A cell membrane or the plasma membrane. So we'll talk much about this. So for you to have a better understanding about it, I'll use this stick model to explain what I'm trying to communicate to you. So for instance, someone tells you that you have 2 sticks, get 2 sticks, then also get another bunch of 2 sticks. Then how many sticks do you have altogether? So the total will be 4. So this doesn't mean that the what is 2, these are sticks. The number, in other words, they're just saying like, yeah, 2 plus what? 2 plus 2. You get it? So this is just helping you to have a better understanding of how numbers works. So same when it comes to the cell membrane, there's a model which we use for you to have a better understanding, which is called the Fluid Mosaic model, which helps you to understand how a cell membrane is. Because once you understand that, it will easily give you an insight of how things do operate. So you need to know that it is a lipid bilayer, meaning that it is made up of 2 layers which are similar. So that is that here which I'm trying to show. So you find that when you just get one thing here, you find that it has a head and the tail. So the tail, it is hydrophilic, then the tails are hydrophobic. So I mean that these tails, when they're even inside, they don't like water. Then the other part, it likes water. So no wonder this, you can just take it as outside of the cell there's a water, then there's inside of the cell the cytoplasm here. So then these are the proteins which are within the cell membrane. So with this understanding, it helps you now to have an understanding of how a cell membrane operates. So now let's go into things now which cross through the cell membrane without difficulties. So one of those substances are non-polar substances, just like such as oxygen and carbon dioxide. They easily cross the cell membrane. These, then as well as smaller uncharged polar molecules such as water. Then when it comes to large uncharged molecules like glucose, they don't easily cross the cell membrane, or charged particles, they don't easily cross. So now how do they cross? Because it is very important for such things to do to cross through the cell membrane. Now what means, or what helps these things to find themselves in a cell? So no wonder they are these now, what we call channel proteins. This is the reason why they are there. These, that's their job. So I'll explain more about these when I come to talk about the types of transport which occur across the cell membrane. So you need to understand, when a cell is transporting things inside or outside the cell, it's either it is using energy or not. So if it is not using energy, no energy is associated, that is passive transport. And those are the types: osmosis, diffusion, and the other. Then the other one, this one, diffusion and osmosis, it is just direct. It is just direct without energy, like this one, direct here, crossing. But when it comes to facilitated, from the word facilitated, meaning that there's something which is helping. So like this one example here, the channel protein here. So that is passive transport, but and using this protein, that's what now it's categorized as facilitated transport. Then when energy is used, ATP, when transporting things, it is called active transport. Then it is also in types. There's vesicles and also protein pumps. Protein pumps, they just behave the same as facilitated. The only difference, under facilitated, no energy is used, but they don't use energy. But for protein pumps, they use energy. So that's a difference. They're similar, but the only difference is about energy. Then when it comes to vesicles, this is what you need to know. So you find that for instance, this thing is about to be excreted outside the cell. So look at the way it is. So the cell membrane need to make itself like that to fold the entire thing, then it releases, it get pinched off. So as you can see here, it has lost certain parts here. Have you seen? It has also reduced in size for it to just do what? Get rid of this thing. So then the other one, this is what is called exocytosis. Then endocytosis, when things are brought in using what is the vesicles. Like for instance here, this example here at my left hand side here, have you seen? Similar, but see, something is coming out inside. Look at it, has lost what? Some part of the cytoplasm. So we can even further go into looking at the types of endocytosis. There are two types. There's the pinocytosis and what? Phagocytosis. Pinocytosis is just water, drinking fluids. Then phagocytosis is all about what? Eating, or when a cell is about to engulf maybe bacteria, but in the same manner like the way I had explained on this diagram here, right here. That is what is happening. So this, I hope it gave you an insight.